Good afternoon, everyone. This is Fei-Chung Chen. I'm an assistant professor from Cleveland Clinical. Today, I'm very glad to co-host this year's tutorial, Artificial Intelligence for Drug Discovery, with my colleague, Dr. Jian Tang and Dr. Fei Wang. First, I will give a very brief introduction for drug discovery, and then I will discuss why we need artificial intelligence technology for drug discovery and development. I think most of you see this drug, discovery pipeline multiple times. So for the entire drug discovery pipeline, first we will focus on the drug target identification, hit and lead compound identification and optimization. If this particular, the lead compound passed the preclinical safety and efficacy, assay, this molecule will go to the phase 1 trial to further confirm or test this drug safety. If the drug passed the phase 1 safety testing, the drug will go to the phase 2 and phase 3, clinical benefit and efficacy testing. If the drug show, significant clinical benefit for a particular, disease compared to the placebo control, we can submit this drug to FDA regulatory department for drug approval. What we learned a lot for the drug discovery process in the past few decades is first in-class drug discovery is time causing. It's, will cost about 15 years and highly expensive. It will cost about $1 billion to $2.6 billion. This figure was released by FDA, every year. This figure Y-X just show the number of drugs approved by FDA across each year in, since 1993. What we learned from this figure is, you can see here, compared to, decades ago, like 1996, 1997, the drug approved by FDA in the past few years doesn't significantly increase compared to 2 decades ago. People argue that we used new technology, experimental technology. We spend more money for the drug discovery. However, the drug approved by FDA doesn't significantly increase compared to decades ago. We see some increase just recent year, but this drug is more like a biology drug. This drug, for example, is a cancer immunotherapeutics agents approved by FDA for cancer therapeutics. So people argue reason why, what is the reason for the drug discovery failures? Here, just summarized some statistical data from the big pharmaceutical company. For example, what we found is majority drug are failed by more than 15% is by no clinical benefit when they see the phase 3 trial. One big problem people see here is most of the drug target is wrong when they do the clinical trial study. The second, failure is the safety issue. Most drugs are failed in the phase 3 trial because this drug have, high toxicity in human. When we look at the different disease area, we see cancer drug is more challenging. I will show more data later. A second, challenge area is we call central nervous system, drug discovery. This, slide just to show the paradigm shift of drug discovery and clinical trial. Like just several decades ago, we just do the symptom-based drug discovery, try to identify the medication. This medication can relieve or reduce the symptom. After that, the physician and the patient argue that, no, we're not just treat the symptom. We have to treat the disease or cure disease. We call disease-modifying therapeutics. Today, we test the drug clinical benefit, use the largest scale cohort-based clinical trial or randomized clinical trial, and then we quantify the average and mean response for a particular molecule in, larger scale, patient cohort. However, what we learn is most of the patient is highly heterogeneity, and most of the molecule doesn't show good mean or average response for this large scale, heterogeneity population, and many drugs are failed we see in clinical trial. People argue, however, this failed drug may also show we call exceptional responder. Good for a small particular population. This is called exceptional responders. And people argue tomorrow or future drug discovery, we really need to focus on what we call precision medicine drug discovery or clinical trial. Precision medicine requires different type of clinical trial that focus on individual drug response, not average mean response to therapeutics. Why? Let me give some data. The figure A just show the success rate for the drug from the phase 1 to get a final FDA approved. But when we look at the cancer oncology drug discovery, only 5% of success rate means 95 drugs into the clinical trial are failed, for cancer drug discovery. For other disease area, the success rate is also only about, 11%. However, when we see, we call the biomarker guideline clinical trial, the success rate can significantly increase to 26%. This is a huge increase. But when we look at the cancer, like oncology drug, like 2016, 0.5% FDA-approved cancer drug are personalized drug. What means the personalized drug? The left figure just shows this personalized cancer medication particularly target a mutant protein, or they have particular biomarker like gene expression to guide the patient selection for this medication. Because we know that each patient is highly personalized in the heterogeneous population. This can, personalized medicine idea really bring the idea we call the big data idea for the drug discovery. We not just identify the drug can treat a human disease, we really want to identify the right drug with the right dose, for a right patient based on the genome information for a particular patient. However, there are many, many challenges for this precision medicine drug discovery. For example, the big data challenge we see from the human genome study. Do you know how long for the human DNA in our body? I just give a number we call the trillion meters. How we calculate this trillion meter? Because based on the single cell study, we have a trillion cells in our human body, and each single cell, the DNA about 2 meters. What does this distance mean? This distance equal about 666 round trip between Earth and the Sun. It's not a big data challenge for the genome and a human biology study. We also have challenge for the chemistry study as well. For example, we have a huge chemical space. For example, based on in silico study, we have hundreds of billion small molecule space. We have to search a particular, molecule for a particular drug targeted treatment. So this is really, the new technology or good model to search the molecule in this huge, space. The second challenge we call human druggable genome. Right now, based on the data, we have about 1,000 human protein can be targeted by small molecule or antibody. They here just show the landscape of human druggable genome. However, we know many drug target is undruggable, like a transcription fighter. This also brings a new challenge for the drug discovery. However, the human protein or drug target is not just isolated in our human cell. They have many interactions we see here. For example, each dot we show here is just to show the human protein, and the connection just to show the protein-protein interaction. So, however, we have many interactions here. We're not just interacting in protein, we also have interaction with the human DNA, RNA, and DNA variation as well. Also have interaction with the metabolites in our human body. So drug discovery and healthcare generate detailed data. This data is critical to create a learning health system for drug discovery and development. However, we really need innovative technology and algorithm to deal with this big data, to bring the power of picker-based big data for the drug discovery. For example, this is Nature Review from 2018. Bring the concept is for use the artificial intelligence approach to accelerate drug discovery process. The idea is we can build the AI model, use the big data we generate. We can collect from different results. Use this AI model can significantly save the time and money for the drug discovery. For example, these people proposed the idea for the drug design in the AI area. For example, we can use the computer to design a molecule, and then we can use the robot, right, to synthesize this molecule just in a few days or few weeks, not in a few months or years. And then we can test this molecule in the in vitro system. If we get successful, we can move this molecule to the clinical trial very quickly. So AI-based drug design and drug discovery can significantly reduce the timeline and cost for the drug discovery. There are multiple different types of technology, including the artificial intelligence technology, machine learning technology, and deep learning technology. Also include the natural language processing technology for the drug discovery process. For example, this is a review paper published last year, 2019, introduced the different type of machine learning technology, including the regression model, classification model, and a clustering model or method. And apply this method, you can see in the entire drug discovery pipeline is here. My colleague, Dr. Fei Wang, will give more detailed introduction for the machine learning approach for drug discovery. So summarize, today, in the next 3 hours, we try to cover the different topic, like the artificial intelligence for drug discovery in this tutorial in the next 3 hours by working with my colleague, including Jian Tang and Fei Wang. We will cover I will try to cover how we use the AI and the network approach to try to integrate different type multi-omics data for COVID-19 drug repositioning and combination study. In Jian Tang, we'll talk about the de novo drug discovery study. Fei Wang will introduce how to use the machine learning for the electronic health record data and for clinical trial data analysis. This is the agenda for our today's tutorial. First, Dr. Jian will focus on the de novo drug discovery. I will focus on the drug repositioning and the drug combination study using the COVID-19 as an example. And Dr. Wang will focus on real-world data driven the drug repositioning study. For example, Jian will introduce a different area for the de novo drug discovery, including the molecular properties prediction, molecular design and optimization, and the compound synthesis process. I will cover how we use the AI and the network approach, try to integrate the multi-omics data for COVID-19 drug repositioning and the combination study. And Dr. Wang will introduce how to use the machine learning approach for clinical data analysis. Next, Dr. Fei Wang will introduce the fundamental concept for machine learning study.
Hello, everyone. My name is Fei Wang. I'm currently an associate professor in Department of Population Health Sciences at Weill Cornell Medicine in Cornell University. And it is a great pleasure to introduce this tutorial together with Jian and Fei Xiong. And in this part, I'm going to give a brief overview of the popular machine learning approaches that could be useful in the drug discovery and development process. As Fei Xiong introduced, there are many different kinds of data that will be involved in the drug development and discovery process, such as biological images, DNA, RNA sequences, protein interaction molecules, as well as longitudinal clinical records. Different types of data are usually involved in different machine learning processes, and typically the machine learning problems are usually in 3 different setups. So one is supervised learning, where there are typically 2 stages. In the training stage, you're given a training data set where for every sample you have its corresponding output or target or class labels. The goal is to learn a mapping function that can perfectly map your sample feature representation to its corresponding target. And the second stage is the testing stage, where you use the learned mapping function to predict for a testing sample where I only have the feature representation. I don't know its corresponding target. So how can I know its corresponding target? I can use that function to kind of like predict. One typical problem for supervised learning is that when you have a limited sample size, which means that which usually causes your training data distribution, testing data distribution to be different. Then that makes your learn function that performs well on the training data not necessarily work well on testing data as well. So this causes a poor generalization ability. So that's something we need to consider when we work with especially limited sample size. Another one is unsupervised learning, where we're only given the feature representations of a set of samples, and there's no notion of target or outcome, and the goal is to for example, one typical unsupervised learning problem is clustering, where the goal is to identify a set of homogeneous groups in a sense that the patient within the same group are similar to each other according to their feature representation, while the data samples from different groups are different from each other. So this is kind of like we are learning the geometric patterns from the samples just based on their feature representation. So there's no really notion of supervision. So that's why it is called an unsupervised learning problem. The other is reinforcement learning. So where number one, this is a sequential decision process, and at every decision step, we need to take an action from the action space, and the criteria to guide us to select, to choose which action I should use is really dependent on, if I take that action, what reward I'm going to get, and that reward is really obtained by interaction with the environment. So the goal of reinforcement learning is to learn an optimal decision path that can lead to some final state in a successful way. So all these three different machine learning setups will be involved in certain stages of drug development discovery process, and you will hear more details from our later on contents. And to give a broad overview, so this is an article from Nature Reviews Drug Discovery 2 years ago, where the authors have summarized the typical stages in the drug development process, including target identification and the validation, compound screening and lead discovery, preclinical development and clinical development. And there are application problems in each stage, such as target identification and prioritization, compound design, tissue specific biomarker identification, as well as treatment response estimation. So for different stage, for different problems, different machine learning algorithms must be needed, because there are different kinds of data involved in different stage and different setups involved in different stage. So we need different kinds of machine learning models to solve those different problems according to the different data. And the authors of this paper also summarize typical like supervised and unsupervised learning techniques that are already involved in these different stages, as well as different application problems to analyze different kinds of data. So this is a very holistic overview of kinds of like the landscape, which you will hear more about, especially the recent advances of machine learning algorithms, like especially deep learning techniques, how they are leveraged in solving these application problems involved in the drug development and discovery process. And I'm going to briefly summarize what kind of machine learning algorithms do we have currently, and which can help you to understand later on when you hear different problems, how these machine learnings can be plugged in to the different scenarios. So for conventional machine learning algorithms, I gave some examples here, including both supervised and unsupervised learning approaches. So for classification problems like logistic regression, which is one of the most widely used linear classification algorithm, which aims to do binary classification using a linear model. Another popular approach is called support vector machine. Where the goal is to also separate two classes and at the same time, they define a geometric margin that reflect how much the two classes can be separated out. So the principle of SVM is to maximize that classification margin. But later on, people have generalized the support vector machine to number one, the multi-class case, number two, the nonlinear case, where they use something called the kernel trick to map the data samples into some high dimensional space, such that the nonlinear classification problem in the low dimensional space becomes linear separable in the high dimensional space. And when they map the high dimensional linear classifier back to the original space, that classification function becomes nonlinear. And the third thing is decision tree. The good thing about decision tree is it's a general, interpretability. So because from the root node to the leaf node, it is essentially a group of rules. And we know for biomedicine and also drug discovery and development, how to interpret how the model works, or explain or interpret how the model works, is really important for the domain expert to understand the entire decision process. And also like Bayesian networks, which we're estimating the conditional probability, especially between the different to reflect the relationships between the different vectors, different variables. And based on that, there are lots of causality type of study, which means that, for like classification, we typically just care about, or general supervised learning, we just care about the predictability, which is a lot of the times is just the association type of study. But usually, association may not be enough. We also want to find the causality relationships. Then we may need those probabilistic approaches to help us to perform some causal analysis to figure that out. And for unsupervised learning, such as the dimensionality reduction technique, such as the principal component analysis, where the goal is to look for a linear projection direction where the data has a maximum spread variance. And there's also the TSNE, which stands for the student t-distributed stochastic neighbor embedding, where we want to embed the high dimensional data vectors into the low dimensional space, usually in 2 or 3 dimensional, so that we can see the structure of the data. And the principle behind TSNE is the pairwise relationships between the data samples in the high dimensional space should be preserved in the low dimensional space after TSNE, so that their relative geometric relationships won't change. And also for clustering algorithms, we have like K-means clustering, and we also have hierarchical agglomerative clustering. We see this in all different places. And in recent years, there's a huge success of deep learning techniques in like computer vision, like speech recognition, like natural language processing, and with more and more accumulated data in the drug development and discovery process. So it is also very promising to apply a lot of these deep learning techniques into our drug discovery and development process as well. And here I just listed some typical examples of deep learning techniques, such as feed-forward neural networks, which sometimes also referred to as a multi-layer perceptron, and like recurrent neural networks, which is good for processing if your data is represented as a sequence, such as a natural language, which is usually you can think of as a word sequence, or in drug development process, those molecules can be represented as Miles code. And for patient electronic health records, you can think of the patient records as a discrete time event sequence if you can concatenate the patient records according to their different visit time from earlier to the latest. And also convolutional neural networks, which is appropriate for, I mean, not appropriate, but good at analyzing image features, image type of data, and graph neural networks, which kinds of like extend the convolutional neural networks to general graphs, where typically the traditional Euclidean geometry won't fit, but you need more kinds of like complex geometric tools to characterize the general graphs. And in addition to deep learning, some other important machine learning techniques you will also hear through this tutorial include causality, such as when we want to estimate, let's say if we generate some hypothesis that this drug could be appropriate for that disease, then we want to validate the effect of this drug in real world patient database. Then we really want to understand that, let's say the patient condition is getting better, is really caused by this drug, or some other factors. So then the causal inference techniques needs to be applied here to derive a rigorous evidence about treatment effect, and also interpretability. A lot of the times, especially with complicated deep learning models, the model usually people set in forms like black boxes. It is difficult to figure out what happens inside, and how, for example, you figure out a molecule structure like this through such a complicated model architecture. So how can we effectively interpret the model, like either in like intrinsic way, like we inject attentions, or in the post hoc way, like we do model distillation? These are all important topics and directions in machine learning that is also crucial to the drug development process. And the last one is stability. So we really want to develop the drug. Let's say the evidence developed from one place can also be generalized to other places. This will involves how can we make the algorithm that are stable across different data sets. So you will hear more concrete specifics about all those machine learning techniques in our later on presentations. And next, Dr. Jian Tang will introduce the third part of this tutorial. Thank you.
So hello everyone, my name is Jian Tang, an assistant professor at MILA, AI Research Institute in Quebec, which is funded by Turing Award winner Joshua Benjo. And thanks, Fission, very much for the great introduction on drug discovery. So next, I'm gonna introduce de novo drug discovery. So let's briefly revisit the process of drug discovery. So usually we have a drug target. So most of the time it's a protein or an enzyme. And then in the stage of lead discovery, we're gonna screen millions of molecules, and then to see which molecules can bind to the protein targets. So we call these molecules as lead compounds. And then in the stage of lead optimization, we're gonna first optimize the properties of these compounds. For example, we may want to reduce their toxicity. And by doing this, we are able to obtain a few drug candidates. And these drug candidates will be further sent to preclinical studies. So usually, so these drug candidates will be evaluated or tested on animals such as mouse. Okay, and once they are able to pass the preclinical study, they will be sent to clinical trials. So there are quite a few important problems in drug discovery which can be accelerated through the technique of artificial intelligence, including molecule property prediction. So in this case, given a molecule or a compound, we want to predict their chemical property. And the second problem is molecule design and optimization. So this process is basically the inverse problem of molecule property prediction. So in this case, we are given a property or a set of properties, and we want to find a set of molecules which are able to satisfy the given properties. Okay, so once we are able to identify a designed drug candidate, okay, so the next problem is, are we able to synthesize this molecule in practice? So this is related to the problem of retrosynthesis prediction and reaction prediction. So basically, given the design, the target molecule structure, can we predict a set of like reactants which can be used to synthesize this molecule? Okay, so of course, in this process, this involves the prediction of both forward prediction and also retrosynthesis prediction. So next, we will focus on the first problem, which is molecule property prediction. So molecule property prediction is a very fundamental task in many different stages of drug discovery, including virtual screening and also lead optimization. So there are many important properties for molecules that we want to predict before we send them send them to clinical trials. For example, the binding affinities with proteins, and also the toxicity, absorption, distribution, metabolism, and also excretion. Okay. And now in machine learning, so if you want to make good predictions, so it's usually very important to have a good feature representation of data. So therefore, we must have good feature representations of molecules. And then let's revisit traditional approaches of like representing molecules. So traditionally, molecules are usually represented as fingerprints, or more specifically, each molecule is represented as a binary vector or a frequency vector. So basically, so you can look at this figure. Each molecule is represented as a binary vector, and each dimension here you can see they correspond to a substructure, and then basically each dimension indicates the presence or the frequency of a specific substructure. Okay, and then you can see the fingerprints are usually very sparse and high dimensional because the number of like substructure could be very large. Okay, and then once we have this very sparse and high dimensional vector or fingerprints for a molecule, we can use very classical algorithm, like for example, support vector machines to predict the properties of molecules. Okay, so this is very traditional way to represent a molecule. And later, there are also more advanced methods based on graph kernels to predict the properties of molecules or molecule classification. So the general idea of graph kernels is to measure the similarity between two graphs. Okay, and once we have the similarity matrix between the graphs, so we can use some kernel methods like support vector machine for graph classification. So what is the essential idea of graph kernel? So basically, for graph kernel, as we said, they try to measure the similarity between two graphs, and each graph is represented as a high dimension vector. And this high dimension vector is basically very similar to the fingerprint we just mentioned. So they basically sort of like encodes the frequency of different substructure. So here FG is actually vectors of normalized counts of substructure. And once we have these like normalized counts of substructure for the two graphs, so we can measure their similarity. Okay, so you can see that for the traditional graph kernel method, and for each substructure, they correspond to a different dimension in the feature vector. So therefore, they actually ignore the similarity between different substructures. Okay, so that's why that's actually the motivation of the deep graph kernel method, which is like introduced in 2015. Okay, so what is the essential idea of the deep graph kernel methods? So basically, they want to learn the similarities between different substructures through embeddings. So they try to learn an embedding for each substructure. Okay, so more specifically, they sort of like try to construct a graph between different substructures. Okay, so here is a vocabulary of like substructure. So the size of this substructure is actually less than 5. Okay, and then we try to construct a graph between these different substructure. So more specifically, so we actually construct a graph between these substructures using added distance. So basically, for two substructures, they are connected when one structure can be obtained by either adding or deleting a node from the other substructure. So by doing this, we can construct an undirected graph between different substructures. So once we have a graph between different substructure, so we can learn the substructure embedding. So basically, then in this case, learning the substructure embedding corresponds to learning the node embedding of the constructed undirect graph. So in their paper, they actually leverage the techniques of word embeddings. So what they did is they can conduct a random walk. So basically, by doing this, they can obtain a sequence of substructures which correspond to sentence, okay? And then they can use some word embedding techniques such as like SIBO or skipgram, which were like state-of-the-art methods for learning word embeddings. So by doing this, they can learn the substructure embedding. So their methods actually correspond to, they're actually essentially the same as node embedding methods on graphs, such as like deep work and line methods. Okay, so people can further read the more details if you want to learn how to learn node embedding. So you can read the 2 papers, deep work and line. Okay. So this is more like unsupervised methods for learning the similarities of substructures or learning or evaluating the similarity between 2 molecules. Okay, and recently there's a huge progress of learning molecule repetitions with graph neural networks, and once the molecule has a good repetition, we can make predictions based on this molecule repetition. Okay, so usually these methods are supervised methods. Okay, so they work in this way. So first, we can apply a graph neural network, which usually consists of like multiple layers of graph convolution layers, and then we can use multiple layers of graph neural networks to learn the node repetitions. So once each node has a repetition, and then we can further add a readout function. So this readout function is basically defined on the node repetitions in the last layer. Okay, so this sort of like correspond to a pooling layer. So basically try to pool the entire set of like node repetitions to get the repetition of the entire graph. Okay, and this this readout function could be very simple. For example, we can either take summation or take the average of the whole node repetitions. A very typical method for learning the whole graph of the molecule repetition is called like a neural message passing network. So neural message passing network is a very general framework for learning node embeddings or learning the whole graph repetitions. So actually, almost all existing graph neural networks can be somehow formulated as a general framework of neural message passing. So this is sort of like a framework that defines how different nodes can pass messages, which are essentially vectors. So there are a few important functions in the neural message passing network, including a message function and also a node updating function. Okay, so in the message passing phase, basically we try to pass messages between different nodes. Okay, specifically, actually, we have like 2 procedures. One is called aggregation, and another one is called combine. So for aggregate function, so basically we try to aggregate for each node. We try to aggregate a message from their neighbors. So here MT is basically a message function defined on each edge. So it depends on the node repetition of the source node and the target node, and also the edge attribute. And then we can try to sum over all the message from the neighbors, and then we can get the messages from their neighbors. And this aggregate message will be further combined with the node repetition itself. And then here UD basically is a node updating function which combines the information of the node itself and also the message from the neighbors. So by doing this, we can get a new node repetition. Okay? And we can do this multiple times, and basically correspond to multiple layers of graphing networks. And then once each node has a node repetition, so we can use in different ways to calculate the repetition of the whole graph. So we can use some simple approach, like for example, take a summation of the node repetitions to get the repetition of the whole graph. But of course, there are also some more advanced methods. For example, in this paper, they actually use two more advanced methods for getting the repetition of the whole graph. So one is called like a gated summation. So basically, so HVT is a repetition of node V in the last layer. Okay, so for and then we can define neural network on the node repetition in the last layer. Okay, and then besides this, we can actually define a gating function for each node. So this gating function sort of like defines the importance of each node. Okay, so this is a gated summation. So another technique is based on set-to-set technique proposed by Vinyas et al. In 2015. So this basically they try to define a function to combine the repetitions in a set in a nonlinear way. So this is an essential idea. Okay, I will not like due to time limit, I will not go into details. But people, you can read the paper for more details. So another well-known method for learning the whole graph repetition is called a graph isomorphism network. So this method is also very similar to the neural message passing network, except how it like aggregate the message from neighbors and also combine with the repetition of the node itself. Okay, and for the message from the neighbor, they simply take summations of all the neighbor repetitions, and then this is further combined with the node repetition itself with some additional coefficient, and then the two will be further transformed in a nonlinear way through a multi-layer perceptron. Okay, and then for the read out function, they actually use a very simple function which basically takes the summation. Okay. And actually, when they try to calculate the repetitions of the whole graph, they first do a readout for each layer. Remember that for each node we have a repetition in each layer, so they can do the readout for a different layer, and eventually the concatenate the repetitions pulled from the node repetitions in different layers. Okay, so this is how the graph isomorphism network work in terms of like calculating the whole graph repetition. So there are also some more advanced graph pooling techniques. For example, in the DiffPool paper, they trying to learn the hierarchical structure of the graph, and then they pour the whole graph in a hierarchical way, and the hierarchical community structure are learned in a differentiable way. So there are also another method called like a self-attentive pooling. Okay, so there are many other like recent methods for graph pooling. So So due to time limit, I will not cover more details. Okay, so the graph neural network methods I just mentioned, so they are very effective when we have a lot of data. However, in the domain of drug discovery, one big limitation that we do not have so much label data. Okay, so that's why we have to rely on supervised learning and also self-supervised learning, which basically learns the repetitions from on-label data. And recently, there's a lot of like progress in self-supervised learning, including like natural language understanding and also computer vision. For example, in natural language understanding, so we have methods like a transformer, and in computer vision, so we have a lot of like self-supervised learning methods, such like the scene contrastive learning framework. Okay, and recently we also see a huge there's also some efforts for there are also some efforts on self-supervised learning for graph. Okay, so next I'm going to introduce a few work on this. So the first work on self-supervised learning for graph neural networks, or for learning the repetitions of graph, is proposed by Hu et al. In 2020, which is published in last year's Neurips. Okay, so their model is actually inspired by transformers on natural language. Okay, so here's how the algorithm works. So given the molecular graph, okay, and then for each central node, okay, so they first define a k-hop neighborhood for each node. For example, in this case, k equals 2. Okay, and then and for this k-hop neighborhood, they also define a set of like a context graph. So here's how they define the context graph. So they first choose a value R1 and R2, which actually so R1 is smaller than K. So in this case, R1 equals to 1. So here K is 2. Okay, and R2 is 3. Okay, and then basically they treat all the regions between R1 hop and R2 hop as some context graph. For example, for in this particular examples, they have like three different context graph. For example, this one is a context graph. So this is a connected component. And this is another context graph, and we also have a context graph here. And then what they did is, based on the k-hop neighborhood, they try to predict the context graph. So by doing this, they can learn a graph neural network to learn the repetition of the substructure or the whole graph. Okay, so this is the first type of approach they proposed, which is called like context prediction. So basically, they try to predict different context graph based on ego network. Okay. So another approach is called like attribute masking. So this one is very similar to the transformer approach. So basically what they did is they can sort of like mask some node or edge attributes. Okay, for example, they can mask out the type, the atoms in a specific position, or they can mask out the specific bond type. And then they try to predict those attributes, the node attributes or the edge attributes based on the neighborhood structures. So this is the second type of approach called like attribute masking. Okay. So another method for graph self-supervised learning is called infograph, which is actually proposed in my group. So this is by Fang Yun Sun, who did an internship in my group like 2 years ago. Okay, so the essential idea of infograph is trying to maximize mutual information between the whole graph repetition and also all the substructure repetition in the molecular graph. So the essential idea of using mutual information maximization is that, so we want to ensure that the whole graph repetition can capture the most so most dominate information among all the substructures. Okay, so more specific infograph works in this way. So we first apply multiple layers graph neural networks. For example, we can use k-layer neural networks to learn to obtain the atom repetitions, and then for each atom, we can summarize all the local structure centered at that node by concatenating all the node repetitions, by concatenating the repetition of this node in different layers. So here H5I is basically represents all the repetitions of all the local structure centered at node I. Okay, and then we can further summarize the information of the whole graph by using readout functions on all the node repetitions. So this can be simply take the summation of all the node repetitions. Okay, and once we have the whole graph repetition and also all the substructure repetition, so what we can do is we further trying to maximize the mutual information of the whole graph, and also with each substructure in the graph. So here mu is the index of substructure in the graph, and we can do this for each graph in the training data set. And the next question is, how are we able to measure the mutual information between the whole graph repetition and also the substructure repetition? So we use the JSON-Shallow mutual information estimator, which looks like this way. It looks very complicated, but actually it's very similar or related to the contrastive learning idea. So let me give you a more intuitive explanations based on the right figure. So it works in this way. Suppose we have a molecule, and then by applying the k-layer graph neural networks, so we can have a repetition for each node, and then further by using the readout function, so we actually can get repetition of the whole graph. Okay, and then we can treat the whole graph repetition and also a substructure repetition in the same graph as a positive pair. So this is a positive pair, and then we can treat the whole graph repetition and also a substructure repetition sampled from another graph. So this is the positive pair, and this two is a negative pair. So we can train a discriminator trying to distinguish the positive pairs and also the negative pairs. So by doing this, we can actually estimate a lower bound of the mutual information. So this is how we are able to maximize the mutual information between the whole graph repetition and also the substructure repetition in the molecular graph. And then we actually further extend this infograph idea to the semi-supervised setting. So basically, we try to combine both unsupervised loss function and the supervised loss function, or in other words, we try to leverage both on-label data and also label data. Okay, a simple way to combine the two object function could be we can just use a same encoder to encode to encode the molecule, so that we can predict target them. So we can either predict the label of the molecule, or trying to maximize the mutual information as well. Okay. So for example, we can simply combine them by introducing a hyperparameter lambda here to balance the two object function. However, such a simply combined the two object function could be problematic, because the two object function, they may favor different information. It could lead to the problem of like negative transfer. So therefore, we actually introduce a new method, a better method to combining the two object function. Okay, so instead of using one encoder for the two object function, so we actually use a different encoder for the two object function. That means we have like one encoder for the supervised model. We have another encoder for the unsupervised model. Okay, so And then, so of course, each encoder trying to optimize for their targets. For example, for the supervised model, the encoder are trained to predict the labels of the molecules, and also for the unsupervised encoder, they try to maximize the mutual information, right? But also we need to make sure make the two object function are able to collaborate with each other. So therefore, so we also trying to maximize the mutual information between the repetitions learned by the two encoders. Remember that for the same molecules, by applying the two different encoders, they can have different repetitions. So what we're going to do here is we're trying to maximize mutual informations of the repetitions learned by the two different encoders. Okay. So this is essential idea of like our semi-supervised graph repetition learning framework. Okay, so there are also other work, other very recent work for self-supervised graph learning. For example, there's a paper called like an algorithm called like Grover, which is published in last year's Neurons. So the Grover idea basically extends the current self-supervised learning model by further using graph level motifs. So motifs are informative substructures which are defined by domain experts. So specific, you can see that. And for the whole graph repetitions, besides predicting the local node context, they further try to predict the graph level motif structure. So by doing this, they expect the whole graph repetition can also encode the important motif structure information. Okay. So this is a Grove paper. Another paper is called like GCC. So what they did is they pre-train a graph neural network on different types of graph, including very big social networks, citation networks, and also small molecules. And then for each specific task, for example, for molecular property prediction, they further fine-tune the pre-trained graph neural network with the label data. So by doing this, they are also they are able to obtain better performance by pre-training a graph neural network on multiple different types of graph. And then there are also other directions on molecular property prediction, for example, based on multitask learning techniques. As we said in the beginning, so one of the biggest challenges for molecular property prediction is the number of labeled data, because for each property of each task, so we have very limited label data. So one thing we can do is we can sort of do multitask learning. So basically we can predict multiple properties simultaneously, and then we can transfer the supervision or knowledge across different tasks. Okay, so for multitask learning, they can actually be divided into two different types of approaches. So one is called like a feature-based approaches. So in this category, basically for different tasks, so we trying to share some low-level feature repetitions, and these low-level feature repetitions, for example, so in this case, and this low-level feature repetitions will be shared across different tasks, across different properties, and then they will be further transformed to task-specific repetitions by using some task-specific architectures, right? So by doing this, the supervision can be shared across different tasks or across different properties. So these are feature-based approaches, okay, which try to share some low-level feature repetitions across different properties. So another type of approaches for multitask learning is called like task learning approaches. And in this category, this approach is trying to learn the relationship between different tasks. So in this case, in the case of like molecular property prediction, so we are essentially trying to learn the relationship between different properties, and they use some techniques called like soft parameter sharing. So take this like figure as specific example. So we have two different tasks, okay, and then and for each task they actually use different architecture. And then what they did is they're trying to measure, they're sort of trying to define a regularization term which measures the difference of the parameters of the neural networks. So by doing this, they sort of trying to regularize the parameters of the two neural networks which are used for different tasks. And for more information about the multitask learning or transfer learning for molecule property prediction or in general for drug discovery, so you can read the two papers. Okay, so here is a brief summary of this part on molecule property prediction. So basically, molecule property prediction is very fundamental task for drug discovery. They can be useful in the stage of like virtual screening and also lead optimization. And then in practice, if we want to make effective predictions of molecule properties, it's very important to have effective molecule repetitions. Okay. And a big challenge to learn effective molecular repetition is that so we don't have like enough label data. So that's why we have to try to reduce the effort of like labeling or annotating labeled molecules. And so therefore we can explore different kinds of techniques such as like self-supervised learning, multitask learning, transfer learning, and also maybe active learning as well, which is not covered in this tutorial, but of course it's a very important topic for molecular property prediction. So next, we move to the second problem, de novo molecule design and optimization. So this process is basically the inverse problem of molecule property prediction. So in this case, we are given a property or a set of properties. We want to find a molecule that's able to satisfy the given property. And for de novo drug design optimization, so we will not start from a given molecule, but instead we will gonna explore the entire chemical space to search the ideal molecules. However, the problem is actually very challenging because the search space of the whole chemical space is very huge, which is the order of like 10 to 60 or to 10 to 100 for drug-like molecules. The other challenge is that the molecules are discrete, which are not continuous. Okay. And usually for de novo drug design and optimization, from the point of view of machine learning, so we can divide them into two different tasks. So the first task focus on generating realistic molecules. So we can generate realistic molecules based on deep journey models. So usually we can represent molecules as smiles or as graphs, and then we can use deep journey models like variation autoencoders on autographic models or recent advancement in deep journey models based on normalizing flows. So we can use these methods to generate realistic molecules. And once we are able to generate realistic molecules, the second task is we are focused on goal-directed molecule optimization. So in this scenario, so our goal is not only trying to generate realistic molecules, but we also want to find molecules which are able to satisfy the given properties. Okay. So usually for the search, for the molecule search methods, so we can divide them into two categories. So one type methods try to search in the continuous hidden space. So another type methods will search the molecules in the discrete chemical space. So for the methods searching in continuous space, so they are usually based on those like variational autoencoder based approaches. So what they do is they first try to encode the molecules into a continuous hidden space, and then they can further decode the molecules from the hidden space back to the chemical space. And then once all the molecules are encoded in the continuous hidden space, we can just do the search in the continuous space. Okay, so basically we can assume that the the function of the properties in the continuous space is a black box function, and therefore, so we can use some black box search methods, for example, the Bayesian optimization methods. Okay, so once we already identify an ideal continuous hidden point with Bayesian optimization technique, we can further decode that data point to the chemical space. So this is the first type approach. The second type of approach is search directly in the chemical space, because for each molecule, so we can actually treat them as a sequence of discrete actions. For example, each molecule can be treated as a sequence of nodes or edges. So we can directly search the molecules in the discrete chemical space, for example, based on techniques like reinforcement learning. So next, I'm going to introduce a few different journey models for molecule generation and optimization based on different type of like journey models. For example, based on variation order encoders, autographic methods, or normalizing flows. Okay, so the first well-known method for molecule generation is called like a junction tree variation order encoder, which is proposed by Jin et al. In 2018. Okay, so this model is based on the variation order encoder framework. Okay, so for this method, so instead of like representing the molecule as a graph of the atoms, so what they did is, so they actually represent a molecule as a tree, actually a junction tree, and then each node in the junction tree corresponds to a meaningful chemical substructure. For example, could be a ring or a bonds. So you can see that. So by doing this, they are sort of able to leverage the chemical domain knowledge. Okay, and also the size of the tree is actually smaller comparing to the graph of atoms. Also, specifically, they use the encoder-decoder framework based on variation order encoders. And for the encoding part, so let's say, so we will start from the molecule, okay, and then we can extract their junction tree. So you can eventually, so this molecule can be represented as a junction tree. And for encoder, so actually they use two kinds of representations. So one type of representation is they can encode the junction tree. So you can see that once we obtain this junction tree and further encode this junction tree, so each node of in the junction tree will be represented as a low dimension vector. So this is one type of representation based on the junction tree. So another type of like representation is based on the original molecular graph. So based on the molecular graph, they can first apply a graph neural network to learn the representation of the whole graph. Okay, so eventually the molecule will be represented as with two low dimension vectors. One is based on the molecular graph, another one is based on the junction tree. Okay, and then for the decoder, so they will decode a molecule graph based on the two-dimensional two vectors. Okay, so specifically for the decoder, they will first decode a junction tree. Okay, so which basically determines how different substructure will be connected. So this is the first step. So once we already determine the structures or the structures between different substructures, so we need to further determine in each position of the junction tree which substructure we're going to use. So this actually relates to a structure prediction problem, because for each position we have multiple joins, and then eventually we have to pick a substructure in different positions. So we can sort of define a scoring function to score combinations of different substructures in different positions. So we can find the one with maximum score. So due to the time limitations, I will not go to the details. Okay, so but this is the high level idea. So once we have the encoder and decoder framework, and for the search process, so we can just directly search in the continuous hidden space. So all the molecules are already encoded in the search space. So we can use Bayesian optimization techniques to identify the points which could potentially have the maximum properties. And then once we already identify the optimal points, we can further decode them back to the observation space, which is the chemical space, and then we can obtain the molecules with designed properties. So this is the first paper. So the second paper is called like a GCPN or graph convolution policy network. So this method is more based on orthographic methods or the methods which search in the discrete chemical space. So more specifically, they actually trying to formulate the process of molecule generation as a sequential decision process. So which sequentially add nodes or edges. So this basically can be formulated as a Markov decision process. So their goal is trying to discover molecules which is able to optimize specific properties, and also in the meantime they want to incorporate chemical rules so that they can have they can generate like validated molecules. Okay, so GCPN is a very general model for goal-directed graph generation based on reinforcement learning techniques. They try to optimize domain-specific rewards, for example, the properties of molecules, and also adversarial loss based on policy gradient methods. And then they also try to act in a chemical environment which is able to incorporate domain-specific rules, for example, those chemical rules. So next, I'm going to give a little bit more details on GCPN. So basically for GCPN, they formulate graph generation as a Markov decision process. Okay, and then so for the states, they basically define all the possible intermediate and final subgraphs as all the possible states. For actions, they define the action as the possible modifications that can be used to on each intermediate step. For example, they could be adding a node or adding edge between two nodes. Okay, and then of course we have the transition dynamics between two consecutive like subgraphs and reward functions and discount factor. So we're going to introduce each one by one. And for the state space, in the beginning, we're gonna start the generation process with a simple graph which only contains a single node, which is a carbon atom. Okay, and then so all the intermediate graph will be the current state. Okay, and for actions, so there are different kinds of actions. Okay, so remember in each step we're gonna we want to add a set of atoms to the current subgraph. Okay, and then so we can either trying to connect a new atom Ci to a node in the current graph, or we can just like focus on connecting the existing nodes in the current subgraph. Okay, so this is action space. And for the state transition dynamics, so during the graph generation process, actually, we will incorporate the chemical rules for the state transition dynamic. So we will only execute the actions which is able to follow the chemical rules. For example, if we have an action, for example, if there's an edge, and then after adding this new edge, the new substructure doesn't follow the chemical rules. So in this case, we will disregard this action, and then we will the subgraph will remain the same. That means like all the infeasible actions proposed by the policy network will be rejected, and the states or the subgraphs will remain unchanged. And for the reward design, so there are two kinds of like rewards, one the final rewards and also the intermediate rewards. And for the final rewards, so there are also a few different kinds of rewards, including the domain-specific rewards. For example, the final property of the molecules, and also another reward based on adversarial loss. So basically, we can train a discriminator to distinguish the true molecules and also the molecules generated by the model. And then by doing this, the discriminator sort of like give a score to measure whether the molecules is valid or not. So we can use the discriminator to evaluate the validity of the molecule. So this is a reward defined based on the adversarial loss. Okay. So and for the intermediate rewards, so we can check the validity of the molecules in each step based on the chemical environment, and also in the meantime we can still measure their validity based on the adversarial loss function. So this is the graph, the GCPM method. And later, my group actually also proposed a new method for molecular graph generation based on normalizing flows. Okay, so normalizing flows is some very recent advancement of deep learning models, which has been making huge progress in image generation. So here the graph AF methods, we sort of like using similar idea like GCPM. So we also formulate graph generation as a sequential decision process. Okay, so in the beginning we will actually start from an empty graph. Okay, and then in each step we're gonna generate we're first gonna generate a new item, and then next we're gonna determine the edges between this new item with each of the atoms in the current subgraph. So we can continue this process until at some points there will be no edges between the new atom with each of the atoms in the current graph. So in this case, we will stop the generation process. Okay. So more specifically, so graph AF actually transforms each graph into a sequence of nodes and edges. So we will traverse the graph through the breadth first search order, and by doing this, each graph will be represented as a sequence of nodes and edges. For example, here Z0, Z1 represent the sequence of nodes and edges for molecular graph. Okay, so you can see that for actually for during the generation process, actually for each action, so we actually has a random variable as associated with each action. Okay, so that means actually we can sort of like define a mapping between a set of like random variables with our observations, which are a set of like nodes and edges. And for each random variable in the latent space, they actually can be generated from a base distribution, for example, Gaussian distribution. So then, we actually use an autographic flow method, which basically defines invertible mapping between the latent space and our observation space. Okay, so the good thing about like using an autographic flow to define the mapping between a base distribution and also the observations, which are a sequence of nodes and edges. So the advantage is that because we can define some very complicated mapping between the base and between the base distribution and also our observation. So usually the model has a very high capacity to model the data density. So this is really a big advantage based on normalizing framework. Okay. So another advantage of the autographic based methods is that the training is very efficiency. So in practice, like for training, we usually have to maximize the likelihood of data. And then by using normalizing flows, what we're going to do is, so we can actually use the inward mapping. So we can map the observation to the latent space, and we can calculate the likelihood in a parallel way. So in this case, the training efficiency will be very high. Okay? And the sampling process will remain to be sequential. So for the sampling, so we're gonna sample a set of like random variables from the base distribution, and then we're gonna generate the nodes edges sequentially. So the good thing about the sequential generation process is that during the sequential generation process, in each step, we actually can check the validity of the current substructure, so which can actually guarantee the validity of our generated molecules by incorporating the chemical rules. Okay, so here are some results of our methods on molecule generation. Okay, so we train our model on the zinc 250K dataset, which includes like 250K drug-like molecules. So for these molecules, the maximum number of atoms is 38, and there are 9 total atom types and 3 different edge types. Okay, so here we compare the performance of different models with different metrics. Okay. So, so the validity basically measures whether the molecules are valid or not, and uniqueness basically measures whether the molecules generated by the model are different from each other. So novelty measures whether the molecules are different from the training data. Okay, so here we compare a few different methods, including GCPN, for example, a JTVE and a GraphAF. So you can see that for all the 3 methods, JTVAE, GCPN, and GraphAF, so we actually generate 100% valid molecules. And then all of them can generate very unique molecules, and they're also very, they can also generate new molecules which are different from the molecules in the training data set. Okay, so this is the first task. So here's some examples of the molecules generated by our algorithm, which looks pretty realistic. Okay. So next, we move to the second task, which is goal-directed molecule generation. And then in order to find molecules with desired properties, so we can actually just like fine-tune our policy network with reinforcement learning. So the state is basically current the subgraph, and actions, as we mentioned, we actually have 2 kinds of actions, either trying to add a new atom or trying to add a new edge between the new atom with each of the atoms in the current subgraph. And for the properties, for the reward, so we can actually use similar reward functions as in GCPN. So basically, so we can use the property of the molecules as final reward, and also we can use some chemical validity as intermediate and also final rewards as well. Okay, so here are the results of the molecule optimization for 2 properties. So one is the P-log-P, and another is QED drug likeness. Okay, so we compare also with the JTVAE GCPN. So here are the 3 best molecules searched by different molecules for P-log-P, for QED. So you can see that for both properties, our model is able to achieve the best performance compared to JTVAE and also GCPN. And here's some examples of the best molecules searched by our algorithm. And another type of a scenario is like is constraint optimization. So in this case, instead of like searching the molecules from scratch, so we can actually start from the existing molecules, and then we can make some small modifications to significantly improve the property of the molecules. So you can see that for example in this case, we can make only a few small changes, but we are able to increase the properties of molecules very significantly. Okay, and then there also another molecule generation method based on normalizing flows, which is actually by Dr. Wang's group. So the method is called like MoreFlow. So MoreFlow is different from graph in the sense that so they can generate the molecules in a one-shot manner instead of like generate molecules sequentially. But by doing this, they can still able to guarantee the validities of molecules, which is a very nice method. So due to time limit, I will not go into the details, but please read the paper in more details. Okay. So all the methods I just introduced so far mainly focus on single property optimization. That means we are only trying to find the molecules which try to maximize a single property. But in practice, for drug discovery, in order to find a good drug target, we usually need to find a molecule which is able to satisfy multiple objectives. So these relate to multi-objective optimization or multi-objective molecule generation. So this is a very recent paper by Ginetta 2020, which is published in last year's SMA. Okay, so in this paper, their goal is trying to find a molecule which is able to satisfy multiple objective or multiple properties. So their idea is actually inspired from the fragment-based drug discovery. So what they did is they first trying to find some meaningful substructures or function groups, or something they call it like rationale. So for each property, so basically for each property, they first identify a set of like rationales, and then in order to find a molecule satisfying multiple properties, they try to combine the rationales from different properties into a meaningful target molecule. So this is the essential idea of their method. So let's look at some more details. Okay, so basically the rationale, they call their method as the rationale RL. So the method is composed of like two stages. The first stage is a rationale extraction. So basically in this stage, for each property, they try to construct a rationale vocabulary. So this is the first stage. And then the second stage is graph compilation. So basically, given a set of properties, and for each property, they can sample one rationale, and then the goal is trying to combine the rationales from different properties into a meaningful targeted molecule. So this is the essential idea of the second stage, which is graph completion. So let's look at a little bit more details about the two procedures. So for rationale extraction, they use the technique, the classical search technique based on Monte Carlo tree search. Okay, so let's first define the goal of the rationale extraction. So basically, the first start from a set of like positive molecules, which is used to train the property predictor. So they want to find so basically for the rationale extraction, they want to find a subgraph, which of course is a subgraph of the positive molecules. Okay, and in the meantime, they want to find a subgraph whose property satisfying that their property. So, and for this subgraph, their property is larger than the threshold for each type of property. So this is another constraint. And also the size of the subgraph cannot be too large, and there's a threshold on the size of the subgraph. And also the subgraph has to be connected. So they want to find a set of the subgraph which satisfy these conditions. And these subgraphs are called like the rationales for each property. Okay, and then what they did is they use Monte Carlo tree search methods for the search process. So they essentially what they did is they try to iteratively construct a search tree by deleting the peripheral bonds or rings. So take this example. So let's look at this example. So we will start from a positive molecule, and then in each step, so we can either try to delete a peripheral bond or peripheral ring. Okay, and then they use some very classical Monte Carlo tree search algorithm. So the essential idea of the search process is trying to balance between the exploitation and the exploration. So I will not go into the details due to time, but essentially eventually they will collect all the leaf state, basically all the leaf substructure which satisfy the the threshold of different properties. Okay. So we can do this like for each individual property, and by doing this for each individual property, we can construct a set of like rationales. Okay, and once they construct the rationales for each individual property, they try to construct multi-property rationales. So which is done by merging the rationales of different properties. For example, in this case, we focus on 2 properties, and for each property, we can sample rationale. Okay, and then in order to find the rationales for multi-properties, so what they did is to first find the maximal trying to find the maximal common substructures between the 2 rationales, and then they can superpose the 2 rationales so that their maximal common substructure will coincide. For example, if you superpose this rationale on this one, so you make this common substructure coincide, then you can get this merged rationale. And we will keep this rationale if their property satisfy the threshold for each type property. Okay, so by doing this, we can construct a multi-property rationale vocabulary. And so once we have the multi-property rationale vocabulary, so we can generate the target molecule. So basically, you can formulate this as a conditional deep journey models. So for example, so here you have multi-property rationale, okay, and then you can sample one rationale from this set, and then you can model this conditional probability, basically the probability of the target molecule condition on the rationale, okay, and then you can generate the target molecule. The generation process is very similar to what we introduced in Graph AF or GCP, and basically we can generate the nodes and the edges sequentially. Okay, so this is how we are able to generate a molecule which satisfies multiple properties. Okay. So to briefly summarize, for de novo drug design optimization, we want to find molecules with designed properties from scratch instead of like study from some existing molecules. And then usually this method can be divided into 2 different type of approaches. We can either encode the molecules into the continuous hidden space, and then we can search in the continuous space based on method like Bayesian optimization, and once we identify a good point, we can decode them back to the chemical space. Okay, so another method basically formulate a molecule as a graph or as a sequence of discrete actions, like as sequence nodes edges. We can directly search in the discrete chemical space with techniques like reinforcement learning. And then we can have different kinds of general models for generating reducing molecules. For example, methods based on variational autoencoders like JTVAE, or methods based on autoregressive methods like GCPN, or based on flow methods like Graph AF and MorphFlow. And in practice, besides generating a molecule which satisfy a single property, is also very important to generate a molecule which satisfy multiple properties. So next, we move to the next problem, reaction and the retrosynthesis prediction. So for reaction prediction, our goal is trying to predict the product molecule given a set of reactants. And then for retrosynthesis prediction, which corresponds to the inverse problem of reaction prediction. So in this case, we are given a product molecule, and we want to find a set of reactants which are able to use to synthesize the target molecule. And for both reaction prediction and retrosynthesis prediction, so the existing approaches can be generally divided into 2 different categories: template-based approaches and also template-free approaches. So template-based approaches usually rely on some chemical rules or transformation rules. So these rules basically involve different substructures or function groups. So it works in this way. Let me give you one example. So for example, for this reaction prediction, so if we saw two substructures in the two reactants, then they are likely to be react, and then we are generate a product molecule which contains such substructure. So we can apply this kind of like rules for both forward prediction and also retrosynthesis prediction as well. Okay, so these rules are usually hand coded by domain experts, or they can be automatically extracted with some graph frequent pattern mining algorithms. Okay, so this the first type of approach is based on template or chemical rules. Okay, so the second one is template-free approaches, which is totally data-driven approaches. So we try to develop some algorithms that hopefully can learn the domain knowledge, such as the chemical rules from the label data. Okay. So here is a briefly comparison between the template-based and versus template-free approaches. So for the template-based approaches, the good thing is interval. But the disadvantage that it requires the domain knowledge from domain experts, and then it could be very slow because sometimes there could be like a lots of like templates generated from the data, and then it cannot generalize to reactions outside of the domain knowledge. So that's the advantage and also disadvantage of template-based approaches. But if you look at the template-free approaches, the disadvantage is that, of course, because total-driven, usually the model is not iterable. But of course, the advantage that is because it doesn't involve a lot of like chemical rules, usually is much more efficient in terms of prediction, and also it has better generalization ability, which is able to generalize to reactions on the side of the domains. Okay. So next, let's look at some very classical approaches for reaction prediction and retrosynthesis prediction as well. Okay. So for reaction prediction, let's look at the very classical approach based on chemical rules or based on templates. Okay, so usually for chemical or for template-based approaches, as we just mentioned, so sometimes we will generate a lot of like chemical rules or templates either by domain experts or extracted through algorithms. So the reality is, in practice, given molecule, sometimes we don't know which rule we should apply, because there could be many rules that could be matched to the target molecule. So therefore, in this paper, they basically propose to rank the effective rules with machine learning techniques. So basically, they train a supervised model to predict the potential effective rules for a given molecule. Okay, so specifically, given molecule, they first represent the molecule with a transitional molecule descriptor, for example, those molecule fingerprint methods, and then based on these feature vectors, they further train a deep neural network to predict the most probable reaction rules. So by doing this, they can apply the identifier rules to the target molecules, and by applying the rule, they can identify a set of like reactants. So this is a very classical approach based on templates. Okay, and then later there's a lot of like there's a lot of like template independent approaches based on deep learning. So one very typical approach is based on transformer. So basically, in these approaches, they, instead of like trying to represent each molecule as a graph, they represent each molecule as a Myers string. That means each molecule is represented as sequence. They can concatenate all the strings of the reactants as a long sequence. Okay, so then the problem of like reaction prediction can be formulated as a sequence translation problem. So of course, and then we can use approaches like sequence to sequence, and the very classic approach for sequence to sequence is based on the transformer framework. Okay. Of course, like representing molecules as smiles has some limitation because the smiles is a sequence which cannot capture the complex relationship between the atoms. So therefore, a more recent approach is still trying to represent the molecules as a graph. So in this new paper, they actually still represent molecules as a graph, and then they formulate the chemical reactions as a sequence of graph edits. For example, so they define a set of like graph actions, including like changing the adding or remove edges between two atoms, adding a new atom to the graph, removing a new atom from the graph, okay, or stop the adding process, okay. So by doing this, they're able to formulate chemical reactions as a sequence of like graph edits on the reacting graphs, okay. And then the model can be trained with maximal likelihood. So basically, given let's say 2 reactant graph, they just need to predict a sequence of like graph edits. So this is for reaction prediction. So here's some experiment results on reaction prediction, and for the data set, so there's a very well known or widely used benchmark for reaction prediction, so USPT or MIT. So this is a standard forward synthesis benchmark which contains around like 400 around 500,000 atom mapped chemical reactions. So here shows some results of the transformer based framework and also the graph edit based framework. So you can see that for both methods, they are actually achieve very good performance, and the 2 methods sort of like achieve comparable performance on this data set. Okay, so this is for reaction prediction. So next, let's talk about like retrosynthesis prediction. For retrosynthesis prediction, as we said in the beginning, they also include like 2 type of approaches. So one based on template, another based on another is template free. So let's first introduce a very effective method based on templates. Okay, so the key problem for retrosynthesis prediction is the same as forward prediction. So the goal here is still we need to find a good template for retrosynthesis prediction. So therefore, like in 2009, at hand they proposed a method called a conditional graph logic network. So the goal of this graphic model is basically to use new networks to identify the right reaction templates. So basically, given a target molecule O, they want to infer the posterior distribution of the reaction templates and also the set of reactants R. So in other words, we want to model this posterior distribution case. And then, so by using Bayesian rules, we can factorize this posterior distribution into 2 terms. So the first term is basically the probability of the template given the reactant molecules, okay, or given the product molecule. So basically, given the product molecules, which template we should select. And the second one is basically given the target molecule and the template, we want to predict the set of like reactants. Okay, and both these 2 probability can actually be parameterized by a new network. Okay, so let's talk about the first probability, the probability of a template for a target molecule. So we can actually parameterize this probability with an energy-based term. So the energy model can be defined in this way. So here W1 is actually the energy function, depends on both the target molecule and the template. So we can actually parameterize this with a graph neural network. So both the two I think their graph structure. And here we have another prior knowledge basically defines whether the template T match the target molecule or not, because we only want to select the templates which is able to apply to the target molecule. Okay, so this is for the first probability. So the second probability can be defined in a similar way. Okay, so we can define energy function or based on neural network to define the matching between the target molecule, template, and also reactants. So here we also have like a term to basically to determine whether once we apply the template to the target molecule. So whether the generated molecule, the generated reactants match the current set of reactants or not. Okay, and both W1 and W2 they are graphing networks architectures, and the parameters can be optimized with criteria like with objective optimization techniques based on maximum likelihood estimation. Okay, so this is a recent approach for selecting the right template for retrosynthesis prediction. And there are also recently some template-free based approaches. A classical, a very typical approach is still the sequence to sequence approach. So in this case, we also represent molecules as a smice sequence. So that it's not surprising we can apply the sequence to sequence based approaches for retrosynthesis prediction as well, similar to what we have done in forward prediction. Okay. And recently, last year in my group, we also developed a new approach for retrosynthesis prediction. Okay, so in our framework, we actually formulated the retrosynthesis prediction problem as a translation problem from a graph, which is correspond to the product molecule, to a set of graphs which correspond to the set of reactants. So the whole framework can be actually divided into two stages. Okay. So the first stage is the reaction center identification, and the second stage is graph translation. So let's look at the whole framework. Okay, so first, given the target molecule, the goal of the reaction center identification is basically trying to identify an edge in the target molecule. So this edge basically corresponds to the place where the reaction happens. Okay, so once we identify the reaction center, so we're going to break this edge. And by breaking this edge, the orange molecule will be divided into two sub graph. So each graph corresponds to a symptom. Okay, and then, but remember, these two substructures, they are not complete molecule. Then that's why we need to have the second stage, which corresponds to graph translation or graph completion. So the goal of the second stage is trying to complete the symptoms into a complete molecule or a complete reactant. So we can do this like for each symptom in the first step, and by doing this, we can obtain a set of like complete reactants. So this is how the algorithm works. So more specifically, in the stage of reaction center prediction, so we actually formulate this problem as a supervised problem. Okay, basically a binary classification problem. So we first determine whether an atom pair or an edge is a reaction center or not, based on the following criteria. So basically, we found the two, we can find a bond between the two ions in the product, but there's no bond between the two ions in the reactants. Then we think such kinds of like a reaction, such kinds of like atom pairs is a reaction center. Okay, and then we can formulate the problem as a supervised classification problem. Basically, we can use a graph neural network to learn the reputation of each edge, and then further we try to predict do a binary prediction whether each edge is a reaction center or not. Okay, so this is the first step. So second step is basically a graph translation. Basically, we try to translate incomplete symptom to the final reactant, and then we use we propose a variation of graph to graph translation framework. So here we actually introduce a latent variable z, so which is used to capture the uncertainty during the translation process, because sometimes for the same symptom, we can actually potentially translate to a different reactant. So that's why we want to introduce a latent variable to capture the uncertainty. So here are some results of retrosynthesis prediction on the benchmark dataset UCPTO 50K, which is pretty much similar to the data set of forward prediction. Okay, and here we compare different kinds of methods, including template-free and template-based approaches. So you can see that for the template-free based approaches, so all graph-to-graph framework is much better than sequence-to-sequence, which is not surprising because, as we mentioned, if we represent molecules as a SMILE string or as sequence, the sequential structure cannot capture the complex relationship between the atoms. But the graph repetition, the molecular graph basically can capture the complex relationship between the atoms. Okay, so this is the template-free based approach. And for the template-based approaches, so the GLN, which the methods we just introduced, so they achieve the best performance. But of course, as we mentioned, like for the template-based approaches, one big limitation that on large data set, the number of template or the number of camera rules could grow very fast, and which will become very inefficient. Okay, and then if you compare our G2G framework with GLN framework, so you can see that our template-free based approaches is pretty close to the performance of the best template-based approaches. Okay. So this is the end of the part. So I also put the most important references for each problem, including like molecular property prediction, reaction prediction, reaction synthésis prediction, de novo molecule design, and optimization. So thanks very much. So if you have any questions, feel free send me an email. And next, my colleague Fei and also Fei Xiong will introduce another topic of drug discovery, which is like drug repurposing. So in that scenario, instead of like designing new drugs, we will try to identify existing approved drugs for new disease. So thanks very much.
Hello, everyone. I'm very glad to see you again. I'm Fei-Xiong Chen. I'm assistant professor from Cleveland Clinical. In the next half hour, I will introduce our lab recent work, how we use the artificial intelligence approach for COVID-19 drug repositioning and drug combination design study. I know any competing interest need to close. Before my presentation, I will briefly introduce my lab. So my lab at Cleveland Clinical, we use the system biology and the network medicine technology to predict the drug target and understand the mechanism of human disease. We use the network-based approach to predict the drug target and design the drug, like a drug repositioning and a combination for a variety of human disease, including the COVID-19. The approach we use, we call the human interaction approach, network approach. In this video, each dot just shows human protein, and a connection just shows the protein, physical protein-protein interaction. The network, human interaction network we use right now at my lab include about 350,000 physical protein-protein interaction, cover about 17,000 human protein in a variety human experimental assay. So my lab focuses on developing new network-based and AI algorithm, try to integrate the different type of omics data, including the genomic data, transcriptome data, proteomics data, to identify the disease network, use this disease network to better understand the mechanism of human disease and identify the drug target for human disease therapeutics development. So this will just show the agenda for my today's presentation. First, I will give a very brief introduction about the COVID-19, and then I will introduce 3 specific projects, how we use the network-based approach and AI approach for COVID-19 drug repositioning and drug combination design. Finally, I will give very brief introduction about our future work for precision medicine drug discovery in COVID-19. This figure just shows very basic biology for COVID-19. What we learned from the past few months in the last year, what we learned is a few key host factors like ACE2, furin, or TMPS2 played a very important role during the SARS-CoV-2 causing agent, for COVID-19 infection process. However, subsequently, a similar lab from the UCSF identify we call SARS-CoV-2 virus host intactome. The left figure just published in the May or June in Nature. What they show about more than 300, virus host interaction between SARS-CoV-2 in the with a human intactome. And the middle just show protein post-modification we call phosphorylation, for SARS-CoV-2 intactome. And the right figure just show the pan-SARS-CoV-2 intactome, means this show the variant host intactome for a different SARS-CoV, SARS variants, including SARS-CoV-1, SARS-CoV-2, and then MERS as well. My lab is trying to develop an AI and a network-based multi-omics approach for COVID-19 drug discovery. For example, we try to integrate SARS-CoV-2 interaction data, transcription, and proteomics data generated from the COVID-19 positive patient. We use the human interaction network model we introduced before, and then we use the real-world patient data like a COVID-19 registry database for validation. So what we learned a lot from the last year for COVID-19 is COVID-19 really a complex disease we call multi-comorbidity for COVID-19. COVID-19 not just pulmonary or problem. They also have caused multiple problems or organ issue, including the heart problem, heart damage. Also, new data show the autoimmune and neurology damage as well. For example, Yadi is a senior data scientist from a lab. Yuan is a senior postdoc from a lab. They demonstrate that use the network proximity approach. They show the SARS-CoV-2 variant protein or SARS-CoV-2 transcriptome or proteomics data to show that SARS-CoV-2 pathway highly overlapped with the different disease pathway we highlight here. Overlapped with the cancer pathway, overlapped with the heart disease pathway, also overlapped with the neurology pathway. This means the drug approved for cancer or other different disease study may potentially treat the COVID-19 as well because the SARS-CoV-2 shared the pathway with the different human disease. Dr. Paul Ehrlich is the father of pharmacology study. He proposed one disease, one gene, one protein, and one drug concept in almost 1 century ago. This concept get very successful for multiple simple human disease treatment. However, what we learned a lot from past few years, especially for high throughput, experimental assay, we found that, or we show that the drug target is not just, FDA approved drug, not just target the single protein. Every approved drug can target the multiple protein. In this, drug target network, the circle just show FDA approved drug across the different categories like cancer or cardiovascular drug. And the square just show the human target. What we show here is FDA approved drug targeted multiple protein. Based on in silico approach, each FDA approved drug can target average about 32 human target in our human cell. Combine all this knowledge together, my lab work on the concept or the idea is we call network medicine. Network medicine is a new research field. Try to redefine human disease and therapeutics, use the system biology and the network science approach. Network medicine provides an innovative way to identify actionable drug target and therapeutic approach for human complex disease. The idea is for a human interaction, we can identify the disease module. For a different drug, we can quantify the drug target network distance with the disease module, and then we can better understand the drug we call on-target therapeutics effect or off-target toxicity effect. So my lab in the past few years or we demonstrate that we can use the drug repositioning idea to treat the human disease in a variety of human disease, including the cancer like a lung cancer or brain disorder or the new COVID-19 crisis we see in the last year. So today I will mainly focus on how we demonstrate or how we use the AI approach or network-based approach for drug repositioning or drug combination design. Just use the COVID-19 as an example. The idea we use for COVID-19 drug repositioning we call network-based host targeting therapeutics. The idea is, I mentioned before, we show that SARS-CoV-2 virus host interaction are highly overlapped with the human interaction. The drug approved by FDA for other disease, this drug may also potentially block interaction between the virus and host interaction. This drug may potentially treat the SARS-CoV-2. Use this concept, we use the network proximity approach. I mentioned before, the network proximity approach, the idea is in the human interaction I introduced in the video we showed before, for a particular disease like COVID-19, we can identify the disease module. For a different drug, we know the drug target, so we can quantify the drug target network distance with the COVID-19 disease module. If this drug target is close with the disease module, this drug may significantly, associate with the disease. So use this approach, we found we highlight here is about top 16 drug. This drug was identified from our 2000 FDA approved or clean drug. This 16 drug may potentially treat the COVID-19 based on host targeting therapeutics concept. In the next few minutes, I just focus on two drugs we are focused on right now is tauromethane and melatonin. So tauromethane is an FDA-approved selective estrogen receptor modulator for breast cancer treatment. So, well from a lab, a senior postdoc from a lab demonstrated that use the computational modeling approach, found that tauromethane may potentially block the spike protein. And multiple experimental assays show that tauromethane have about a micromolar concentration for a few, the variants, different variants include the Ebola, SARS-CoV-1, MORAS, and SARS-CoV-2 as well. This shows tauromethane a potential, small molecule for the COVID-19 treatment. The second question we wanted to ask, how we can, validate the drug efficacy or clinical benefit. So we use the idea is we call real world data. Use the propensity score approach for the cohort-based design, case control design. We really thank you, my collaborator, my colleague, Dr. Laura Jehai at Cleveland Clinical. We built the largest scale COVID-19 registry database at the Cleveland Clinical, covered more than 15,000 patients. Each patient have, SARS-CoV-2 outcome data, and each patient also have multiple medication data. Use the propensity score calibration design, we can test the drug efficacy use this registry database. My colleague, Dr. Fei Wang will introduce more, advanced machine learning approach for the real world data, study. So what we show here is what we found interesting is use the propensity score match approach. We will justify different disease, different confounding factor, include aging, gender, race, smoking, and disease comorbidity. We found is if individual take melatonin, active take melatonin during the SARS-CoV-2, RT-PCR testing, we found this individual can reduce about 13% likelihood of SARS-CoV-2 positive. So this means melatonin may have potential, treatment effect or prevention effect for COVID-19 patient. The question comes out of why? Because melatonin is a very magic molecule. They have about 2.5 billion history for melatonin in our world. We can find melatonin in different organisms, including the bacteria, virus, animal, and our human health. So now melatonin is a supplement we can easily buy from the Costco or other supermarket. to help us to improve the sleep disorder. So melatonin is targeted the circadian rhythm, system in our human body. So when we look at the COVID-19 patient, transcriptome data, what we found interesting is the circadian rhythm gene are also just, differently expressed in the COVID-19 patient compared to the health, subject as well. These also show, potential or possible, prevention effect or treatment effect of melatonin in COVID-19 patient. The second project we work on right now is AI-based COVID-19 drug repositioning. So this project come out from the idea is from the forced network proximity study, depending on the drug target network we use. However, we know most effective drugs have missing target. This drug may have more target, but we don't know based on the limited data we can get from the experimental assay. We think about, could we identify the COVID-19 drug target, use the AI-based approach? So the approach we use is we call the open data science approach for COVID-19 drug discovery. The approach is we take the largest scale public available data, like the 24 million research article we can get from the PubMed. We use this largest scale, the open data to build the knowledge graph for COVID-19. With this knowledge graph covered, comprehensive, relationship among drug, gene, and the disease. This knowledge graph, COVID-19 knowledge graph, include about 39 types of relationship between drug, target, and disease, cover about 15, over 15 million each. We use the ROTE, approach, deep learning approach to build this comprehensive knowledge graph, and then with systematic way to predict which drug may have high, predictor score for COVID-19 treatment, and then we select this top predictor drug, use the bioinformatic approach to do validation to see if this drug can reduce or induce the transcriptome change in the COVID-19 patient when we're mapping the drug-induced transcriptome data with the COVID-19 positive patient transcriptome data to do the bioinformatic validation. To combine, AI-based prediction and the transcriptome-based validation, we identify, about 40 top drug candidates. This drug candidate include the drug we mentioned before, include the melatonin and the tauromethylene we identify use the network-based approach. I want to highlight another drug is very top 5 prediction we call dexamethasone. So after our paper published just 2 weeks, we see dexamethasone was approved by the UK, and now they also use it in the US for hospitalized patient treatment, hospitalized COVID-19 patient treatment as well. After that, our paper just published as a cover paper by the, the GPR. This work also get some, released by a few, website to highlight our work, how we, to demonstrate use the AI approach can accelerate, the COVID-19 drug repositioning study. The third project I wanted to highlight, mention some question for COVID-19 treatment is, for example, when we know that remdesivir is the only drug approved, in US for COVID-19 treatment. However, remdesivir only show very limited clinical benefit for COVID-19 treatment. So another question we think about, could we improve the COVID-19 drug treatment? The idea we use is we call drug combination idea. Drug combination idea is we try to combine the 2 drugs or multiple drugs together to fight or to treat the complex disease. This kind of concept get very successful for complex disease treatment like cancer and HIV virus as well. The idea is we know the monotherapeutics, we use the high dose that have high toxicity and drug resistance. Drug combination uses the low dose for multiple aging or multiple medication together, combined together. This kind of drug combination strategy, they can have high clinical efficacy, but a low toxicity and a low drug resistance. Our idea for drug combination design is for the human intactome, we know the disease module. And for different drug, we know the drug target. So our idea is, could we look at the different drug target network relationship with the disease module, and then we can predict the drug target. So the approach we use here is I published, in Nature Communication 2019. What we demonstrate very interesting is in the human intactome for a particular disease, if two drugs are overlapped with the disease module, but these two drugs also overlap each other, this kind of we call overlapping exposure is not good for combination design. This kind of drug combination have good efficacy in preclinical study, but they often have high toxicity when we use in the clinical or in the clinical trial. What we show here is only two drugs are close with the disease module. These two drugs not overlap each other, we call complementary exposure. This type of drug combination have high clinical benefit, but low toxicity for human disease treatment. And another C and D, we call single exposure or no exposure. We doesn't see the good clinical benefit when we look at the clinical data. So use this, complementary exposure, design, we found the melatonin and tauromethrin is the best drug combination candidate, we found from our prediction for potentially treat the COVID-19. This just to show, how we quantify use the network proximity approach to quantify the relationship between the drug and drug with the disease. Now, based on this finding, we have initiated the clinical trial to test the melatonin or melatonin combined with the tauromethrin in potentially treated the COVID, especially work on the earlier COVID-19 patient at our Cleveland Clinical. Our IND also get approved by the FDA. Our IB also approved. We're also waiting for the patient recruitment now to study this trial with my clinical the colleague, including the Rina and the Sujeta. Just give brief summarize for my today's presentation. Here we just show example how we use the network-based approach and the AI approach to accelerate COVID-19 drug repositioning. The idea is we try to integrate the multi-omics data to identify the drug target or mechanistic pathway in SARS-CoV-2, and then we use this, the network to systematic way to screening the drug. If this drug come out of our top network-based prediction, and then we use the large scale real-world data for the drug efficacy validation, use the clinical data. If we get successful, we can quick to move this molecule for the clinical trial testing. So this kind of strategy, can significantly reduce the timeline for drug discovery. This kind of strategy, have good advantage for the pandemic, especially for COVID-19 pandemic and our future, the passage, emerging passage pandemic as well. I just want to discuss a few results. We also get very interested is from our the COVID-19 registry database validation. For example, what we found is melatonin can reduce about 15%, 50% likelihood of SARS-CoV-2 positive rate in Black Americans, but we didn't see highly significant clinical benefit for white Americans. We also do the subgroup analysis for melatonin in different the patient group. For example, we look at the individuals have asthma or individuals have diabetes or hypertension. What we found interesting is melatonin can reduce 15% likelihood of SARS-CoV-2 in diabetes patient, but we didn't see significant clinical benefit for asthma patient or hypertension patient. So this data combined together to highlight the importance of precision medicine drug discovery for human disease, including the COVID-19. So right now we are working very hard to work on the COVID-19 precision medicine study. For example, we are working on together with the Broad Institute to do the whole genome or whole exome sequence study for the COVID-19 patient at our Cleveland Clinical. We try to apply the network-based and AI approach to identify the unique disease pathway. And then we use this unique pathway to identify the drug. This drug may only have good clinical benefit for this particular patient. This patient carrying, particular genetic variation or have, particular, the genetic biomarker or expression biomarker. Finally, I really wanted to thank you, my lab member, especially for, the Yadi, Yuan, and, they focus on this project. I want to also thank you, my colleague, Jian Tang and Fei Wang for, to co-host this, tutorial, and for our other, ongoing cooperation project. I want to thank you, my collaborator include, software algorithm, my chair, Karis In, and Laura Jihai for the coordinating registry database collaboration. I really thank you, my clinical collaborator, including Reina Mohai and Sujita Rao for, the clinical trial collaboration. I want to really thank you, the funding support from the NIH or other, the funding agency. Finally, I will really thank you for your attention, and we try to answer your question after, after this, the video. Finally, I really wanted to post some job here. We have some few, postdoc job open, use the AI and the network-based approach for the drug repositioning or drug discovery study. Feel free to reach me if you're interested in my lab or my lab research project. Thank you for your attention. And next, I will move to my colleague, the Dr. Fei Wang. Dr. Wang will introduce how we can use, the machine learning approach for, to analyze the larger scale real world data for drug repositioning study, or, validate the drug, disease, outcome, use the real world data. Welcome, Dr. Wang.
So Fei Xiong has introduced his work on using multi-omics and knowledge graph for computational drug repurposing, especially on the disease of COVID-19. So now I'm going to introduce how the real-world data, such as patient longitudinal patient clinical records, as well as claims, can be leveraged or used in the drug repurposing process. So computational drug repurposing, we know there are two types of main modules. So one is hypothesis generation, such as you can generate from multi-omics or knowledge graphs. And from this part, you can also hear that they can also be generated from real-world data. But another one is how can we verify or collect the evidence that support this drug could be an effective treatment based on your hypothesis. So we have this two parts. And so real-world data mostly has been leveraged as a evidence generation resource to produce the necessary evidence, especially on human data, for supporting the hypothesis. So real-world data, it is like typically when we talk about real-world data, we mainly refer to like longitudinal clinical records or the longitudinal pharmaceutical or insurance claims data. We call them real-world data is because these data are collected from real patients, while as a comparison for clinical trial data, I mean those patients that are recruited to specific trials, they need to have very rigorous clinical trial eligibility criteria. The reason why we want to have those stringent eligibility criteria is because there are so many what is so-called the confounding factors, and which can generate false positives like this, not just the false positive, but false evidence on whether it is really because of the effect of this drug this patient is getting better or worse. So these eligibility criteria is really trying to, balancing out the effect of those confounding factors that really generate reliable supporting evidence. So in real world data, unfortunately, those patients can have many potential confounding factors, and therefore, so how can we mimic the clinical trials to make the evidence really reliable is a key problem for people studying how to leverage real-world data to study treatment effect or emulating clinical trials. So the evidence generation process typically include 3 steps. So one is cohort construction, one is feature extraction, and the last is treatment effect estimation. So in the later part of this of this section, you're going to see how those 3 are performed by existing works. So first, I want to introduce this a fairly classical work on validating drug repurposing signals with here, I mean, it's a EHR that's leveraged in this study for verifying that metformin could be beneficial to like reduce the cancer mortality. And in this paper, I mean, the authors did not really study the causality relationship like it is because metformin that cause the cancer mortality reduction, but it's really an association relationship based on the survival analysis. And this is the standard definition and brief description of drug repurposing that this paper studied, and they specifically emphasize in this paper they are studying the association. And this paper leveraged 2 large EHRs. So one is from Vanderbilt Medical Center, the other is from Mayo Clinic, and they identified about excuse me, 80,000 cancer patients, and they identified among those patients, some of them are with type 2 diabetes with drug exposure, some of them not. And then they use stratified Cox regression hazard model, which is a popular survival analysis model to study like what is the association between taking metformin and the cancer survival rate. And they, to balance, as I said, there could be a lot of confounding factors. To reduce the effect of those confounding factors, they adjust the estimation with age at diagnosis, sex, race, body mass index, tobacco use, insulin use, cancer type, and non-cancer Charlson comorbidity index. And this is the result, and their observation is that metformin was associated with 22% decrease in the overall mortality compared to other oral hypoglycemic medications. So which means that, I mean, although they are with type 2 diabetes, metformin is not the only drug. They also compare the effect of not the effect, the association of metformin with other type 2 diabetic drug. And they find metformin was also associated with a 39% decrease compared to type 2 diabetes patient on insulin only. And the conclusion is the EHR data suggested that metformin was associated with decreased mortality after a cancer diagnosis compared to diabetic and non-diabetic patients not on metformin. So this is their study design, or sometimes what we call this inclusion exclusion cascade. They first need to define or specify the cancer patients, which is in this study based on ICD-10 code, which stands for International Classification for Oncology of Disease for Oncology, and tumor registry. And for the patients with and without diabetes, that is never like a lot of times we just use like ICD code, but it is a well-known issue that simply rely on ICD code may cause the estimation of the sample size not accurate enough we also need to leverage other information like medications along with the clinical notes. So this paper, they identified patients with and without diabetes using 3 types of clinical information: the ICD code, the medication, and also the clinical lab result. So this makes their cohort more precise than just to rely on ICD code. And they also explain to identify the drug information like the metformin. They use both structured and unstructured information. Here, unstructured information mainly refers to the clinical notes. And in order to confirm the patient is on the continuous use of medication, they require the patient to have at least two mentions of diabetes medication in the EHR. And at least a mention within 5 years after their cancer diagnosis date to classify the object by the classification. Metformin medication included in monotherapy and combination therapy are all included, and cancer patient without type 2 diabetes were included as an additional unexposed comparison group. So we can see it is carefully designed. Clearly, this is a collaboration between clinical informatists and the clinician that come up with all those rigorous definitions about the cohort, the drugs, the exposure and non-exposure group. And here I just put the inclusion and exclusion cascade on Vanderbilt and Mayo side by side, so you can compare. I mean, they pretty much follow the same protocol on both sides. And this is the descriptive characteristics of the cohort on both institutions. We can see they are pretty similar. And this is actually the Kaplan-Meier curve, survival curve of the patients, of the cancer patient who are non-diabetic or on metformin or on other drugs or on insulin only. And from the curve, we can see that for patients who are on metformin have a clear higher survival rate within 5 years after they diagnosed with cancer. So this is the association they obtained from the stratified Cox regression model. And this is the examining on the robustness of the discovery with respect to different cancer types. So we can see that for patients with metformin compared to on other drugs, I mean, patients who are cancer patient with type 2 diabetes, we can see almost on all cancer types, the effect of metformin in terms of the association with survival rate is higher compared to other type 2 diabetic drug or insulin. And some interesting extension the authors have done is, okay, so for this study, it is a clear cohort study and with a very careful protocol design, and also all the variables like they controlled for confounding effect have been adjusted and selected in a handcrafted way. I mean, the whole study is just based on a single drug, which is a clear hypothesis-driven study. So can we extend the study to more of like a big data fashion where they consider all the drugs and let's include all potential variants, and this will make this study from a hypothesis-driven to a data-driven study. So hopefully we can they can get more interesting result, and this is a result they get. And the top one drug is something called sildenafil. So for people who are not familiar with this drug, this is essentially Viagra. So I mean, you can see the p-value is 0, which means that for patients who take this drug, they have a significant benefits in terms of the survival rate compared to the patient who didn't take this drug. So so this is kind of like exciting and surprising result. But after consulting with cancer experts, the reason is potentially because I mean, when they do the study, I mean, the cancer, they didn't consider specific cancer types. So there is a specific type of cancer, which is called prostate cancer, which, I mean, for patients with prostate cancer, they tend to have a higher survival rate or longer survival time. And Viagra or sildenafil is actually kind of like a default drug prescribed for patients with prostate cancer. So it is not really, this drug is effective, it's really because they included this specific group of patients who by default gonna prescribe this drug that cause them, and for this group of patients, it tends to live longer. So we can see that without a good study protocol, we can easily get into this scenario of false positive discovery. And the second study I want to talk about is this recent paper from IBM Research, where they use real-world data. In this case, is both the market scan data from Truven and also the EHR data from Explorers. And they want to generate computational drug repurposing hypothesis, as well as evidence for drugs that could be potentially beneficial to Parkinson's disease. And this is their study design, where they first define PD initiation date, and then they set the index date to be the first prescription of the assigned drug. Here, the assigned drug is your repurposing candidate that is generated from the hypothesis. And they define a follow-up period, which starts from the index date until the end of the treatment compliance, which is kind of like the end of the last prescription of the drug during the follow-up period. And they want to leverage the information from the baseline period. Here they set the baseline period to be a little bit earlier than the PD initiation date to the index date. A little bit earlier here in this paper is 6 months, is because the nature of Parkinson's disease is chronic and progressive. So if it is the patient is diagnosed with PD on a specific day, I mean the condition must be already there for a while. And this is an overview of their framework. They have observational healthcare data database, and they have to do hypothesis generation by extracting potential repurposing candidate. And then they, of course, when you define the cohort, the study, and the drugs, you have to come up with a set of user definitions. And then they have a drug repurposing engine, including a causal inference library as the random controlled clinical trial emulator, and then they generate evidence as the estimated treatment effects. So remember, we have 3 steps for the real-world data-based computational drug repurposing. The first is the cohort construction, and excuse me. So this is the different kinds of criteria, from the diagnosis side and also from the drug side. and also from the peer side on how they define an appropriate patient cohort to perform this study. And for feature extraction, so they have built some feature extraction pipeline from longitudinal electronic health records or real-world patient data. So they just leverage that. This pipeline can generate a bunch of, after you define an observation period and also the feature aggregation type, they generate a vector. So the feature needs to be aggregated, but it's not necessarily just the average, but they also calculate some trend features. If you're interested, you can read their paper more in detail. And this is their randomized controlled trial emulation engine, where it's essentially first is they want to come up with some balancing technique to really assure that the feature distribution between the cases and controls should be balanced in order to eliminate the effect of the distribution mismatch or the confounding issues. And they use particularly an inverse probability weighting technique to do that. And here the probability is really the probability of receiving the treatment. And in this paper, they just use some simple logistic regression method as in traditional propensity score method. But of course, you can use more advanced method like recurrent neural network type of stuff to estimate that. And then they test the balance. If not, they just do this for loop again. They also have some adjustment procedure to make sure that after several iterations, you are able to judge if it is balanced or no. And finally, they use two types of ways to estimate the treatment effect. And this is some summary statistics on because in order to evaluate the effect of the treatment, you have to define some outcome events. So here they use some PD related outcomes like dementia force causes or other outcome, and the drugs they have tested for repurposing, for potential repurposing purpose. And they finally come up with a short list of a set of drugs that they have clear evidence that from of course, the real world data they use that can be potentially repurposed to PD. But during this process, this is kind of like a standard, especially statistics driven real world, you can say real world evidence generation engine for computational drug repurposing. And there are several important aspects we need to keep in mind. So number one is define a realistic outcome or clinical endpoints is really important. In this case, the authors just define some events that could be identified by diagnosis code, but in reality, the clinical endpoints could be much more complicated. A lot of the times, the rigorous clinical endpoints used in clinical trials cannot be directly found in real world data. So how to define that? So this is just a table on different kinds of real world clinical and surrogate clinical endpoints. And in cancer, and number two is how can you balancing the data with respect to cases and controls. And this paper used inverse probability weighting, but there's also some other approaches we can keep in mind as well. So, and these are the two studies about if we have a repurposing hypothesis, how can we leverage real world data to validate that and generate evidence. But this is still based on a cohort type of validation, where the treatment effect we estimated is average treatment effect over the cohort. But in recent years, we know the treatment paradigm has been shifted from like one size fits all drug to precision medicine, where we really look at the treatment that are tailored to individual patients. This is because although the patients are with, let's say, with the same disease, their actual conditions could be very different due to the complexity and heterogeneity of the disease. So how can we really estimate the treatment effect for individual patients? So we have done some prior work based on network analysis. We construct a patient similarity network, a drug similarity network, along with a patient drug association network. And then we basically model this, here the Jaccard stands for the Jaccard distance, and SW stands for the Swenson, the Smith-Waterman alignment score. And so the effective of the drug can be viewed as a label on such network, and the patient-patient relationship, drug-drug relationship, and patient-drug relationship can be viewed as an adjacency matrix in such a hybrid network. And the drug label, let's say for a specific drug, could be 1 if this drug is effective for this patient. On the drug side, it could be if the drug is in the network itself or no. And then we perform label propagation on such a hybrid network to predict if the drug could be effective for other patients as well. And we validate this study on hyperlipidemia patients with respect to various studies. And we also have a rigorous design by consulting our cardiologist. And we showed that by considering both patient drug similarities, it can improve the prediction performance compared to if you just consider a single type of similarity. And in this very recent paper on CPT, Mihaela and her group actually surveyed the existing study about how to estimate individual treatment effect from real-world data. On the top figure is kind of like a pipeline where how can you eliminate the confounding issues is still, and estimate the causal treatment effect is still a challenging and a critical issue to address for machine learning models. And the bottom figure just to show, another complication factor is the real world data is also longitudinal. So how can you consider the longitudinal treatment effect of a drug is another important aspect to study, and needs a lot of machine learning innovations. So for people who are interested, you can read their survey paper to get more information. With all that, that's all about this part. If you have any questions, feel free to email me. So we have introduced so far about in silico drug design and development, as well as computational drug repurposing. And we know that the pharmaceutical research and development is a lengthy and costly process. And this figure has shown like the timeline and the cost and the success rate for every stage during drug development and the discovery process. So there are also a lot of other questions and problems that are critical, are critically impacting the efficiency and effectiveness of the entire process. So in this part, I'm going to briefly introduce two other scenarios during this process that machine learning could be very helpful with. So number one is the clinical trial design. So this is a paper published by the IBM folks 2 years ago, and they overviewed the potential of artificial intelligence in the process of clinical trial design. And this figure shows you can think of as almost a vertical version of the figure I showed in the last slide. The success rate as well as the investment or the cost during the entire drug development cycle, from which we can see that the clinical trials occupies almost 50% of the investment that the pharma gonna put into the research and development of a new drug, and the success rate for a drug that pops out after the clinical trials is really, really low. So then how can we leveraging especially real world data to derive data-driven insights that can help us improve or optimize the clinical trial process is definitely something that is not only beneficial to the pharma and from the financial perspective, but also to the patient that can accelerate the drug development process to a significant amount. So in this paper, the authors envisioned or summarize the clinical trial process into different parts, including the cohort composition, patient recruitment, and patient monitoring. And among those different parts, there are different problems like the suitability. The cohort composition determines the suitability of the participants to this clinical trial or the drug study. And also the associated problem includes eligibility, empowerment, and motivation for patient recruitment. And for patient monitoring, we need to closely monitor the drug adherence. We don't want people to take the drug on and off. And also the endpoint detection. We have already touched this concept a little bit in the last section I've introduced about drug repurposing and also the retention. And the methodology involved in those different processes, such as clinical trial enrichment, biomarker verification, trial matching, event logging, in terms of the outcomes, if you remember, and dropout risk forecasting and intervention. And the goal of these different modules, such as reduced population heterogeneity, prognostic enrichment, predictive enrichment, and so on and so forth. So these are kinds of the problems. And during these different parts, there are also different kinds of data that could be leveraged, like EHRs, omics, literature, clinical trial database, Internet of Things, and so on and so forth. And associated with analyzing of different kinds of data, of course, we're gonna have different kinds of AI techniques, including deep learning, like computational reasoning, human-computer interaction, and so on and so forth. So you may wonder, so what is a clinical trial look like? So this is a specific trial about a intervention to Alzheimer's disease on clinicaltrial.gov. So you can see how the study design or the protocol looks like. And all the contents here are actually the eligibility criteria for patients who will be or are eligible to be participating in this trial. So we can see there are lots of considerations from all different aspects, from patient characteristics, demographic characteristics such as age, as to the caregiver consideration, to all the way to the prior conditions and medications history and so on and so forth. So I mean, as I mentioned in my in the previous part section, so the reason why we want to put such a rigorous trial criteria is really we want to like make sure I mean the evidence generated or the treatment effect estimated is really of a high standard that can I mean there's no like false positive or false negative estimations. But as you can imagine, with such a complex structure of the trial eligibility criteria, it's pretty difficult for the researchers to come up with such a list. As you might be imagining that these rigorous trial criteria will severely restrict the participant population, which makes those population quote unquote ideal. So they are not really representative of the real world patient. That's why a lot of the drugs, after they are on market, you still observe this and that a side effect, let's say. This is because the real world patient don't always look the same as the trial patient. So then one of the research question or motivation is, can we leverage real world data to help us refine or design better eligibility criteria such that the patient who are recruited according to these newly improved criteria, they are number one, more representative, number two, suffer less severe side effect or adverse event. Third, I mean, can benefit from the treatment. So in order to do that, we have conducted some preliminary studies. So in order to talk about the representability of the clinical trial criteria, so we first define these 4 different trial populations: the target population, which is actually, you can think of all patients with the disease. The second is study population, so who are eligible based on the eligibility criteria, and the study samples, so who are actually enrolled in the study, and the real world population or the electronic health record population. So who are kind of like the real world patient who were likely to receive the drug. And there are 2 types of ways to measure the generalizability or representativeness of trial eligibility criteria. So one is eligibility-driven generalizability. So it stands for the representativeness of the study population within the true population. The second is sample-driven generalizability. It studies the representativeness of the study subjects and within the true population. So we have conducted a study of the generalizability or the clinical trial eligibility criteria designed based on real world data using the One Florida data, which is an electronic health record data network for the hospital systems in Florida. And it includes like 15 million patients. And we want to, the trial we are investigating is the trial I just showed you on the previous slide, that Alzheimer's disease trial. And after some filtering, we can estimate the numbers within the One Florida data according to those different definitions of the populations. And as we said, so we want the trial, I mean the patient or the participants to be safe on the trial. So we have to define what do we mean by safe. So here we define them as less likely the adverse event gonna appear, and we define the adverse event according to this definition from the common terminology criteria for adverse events, and with the system organ plus defined in medical dictionary for regulatory activities, the AEs can be organized in an effective way, and CTCAE also provides the grading of the AE, and here we consider grade 4 and 5 as a severe adverse event or SAE. So we want, the patient who are enrolled in the trial to have as minimal as possible severe adverse event after they take the drug. So this is actually an investigation. What if we just based on the existing trial eligibility criteria, we call them the study traits, to cluster the patients who satisfy those study traits or eligible for the trial according to the current eligibility criteria. We do a testing embedding of those patients according to their study traits. We can see they are separated by different small cohorts, which is mainly driven by the their age distribution, and we also color them according to if they have SAE or no later on. And actually, figure C here represents a patient who suffered from SAE, severe adverse event later on, and D represents a patient who don't suffer from that. So we can see just based on the study traits, this is complete. Those two populations completely overlaps. So which means that based on the existing study traits, so we cannot really separate the patients who are likely to suffer from SAE or no in the future. And we developed a machine learning technique that by incorporating the SAE as a partial supervision information, so we can derive a t-state embedding with the patients who suffer from SAE and who don't suffer from SAE well separated. So this demonstrates the potential benefits of machine learning model that can really help us to design the eligibility criteria that can really distinguish the the patients who suffer from SAE or no. And another topic I want to talk about is about the drug toxicity and safety. So especially pharmacovigilance, which means that after the drug is already on market, we want to leverage real-world data to build a machine learning algorithm to study if it is likely this drug is going to cause some severe side effect or no. And we have some prior work on studying the association between therapeutic indication and side effect. We want to answer the question like, for drugs that targeting for a specific type of disease, what is the likely side effects they're gonna have? And we build such a relationship. We estimate such a relationship based on simple Fisher exact test, and we build this visualization tool so that the user can look at, the blue and orange indicate the therapeutic indication and side effect. So we can see for certain cohort, you can select of therapeutic indication and side effect. So what are they, so that you can understand the associations as a kind of like a warning signal. And another side effect is caused by drug-drug interactions. So when you take multiple drugs, it's not always A plus B gonna get you the benefit of A plus B, but they can cause severe side effect. We want to avoid that, and we build a drug similarity network, and we assume if one drug interacts with a specific drug in the network, then it is very likely for the drug similar to that drug, this index drug can interact with that as well. So similar to what I have introduced in the label propagation process for personalized treatment recommendation in last section about RWD for computational drug repurposing. We also treat this as a label propagation process on the drug network, and we got we can get some good predictions, and these are the data we have leveraged. We use the two sides, label side effect and off-label side effect database to construct the drug network, and this is one specific example. We discovered like if you are hypertension patients, their to different groups of antihypertensive drugs. And let's say if you also want to take some anti-inflammatory drugs like ibuprofen, then it is very likely if you took like CCBs or CAAs will be more safer. Will be safer compared to if you took those NSAIDs together with ACE inhibitors or ARBs. The numbers on those colored arrows are estimated probabilities, and we also find some clinical studies that supports why the side effect that happens and why it is safer to go the other way around. So this basically shows the power of using machine learning techniques to estimate the drug-drug interactions. And that's all I want to talk about. Of course, there are more topics, and for just these problems as well as other problems. If you have any questions, feel free to email me. Hello, everyone. So far, we have introduced the main contents of this tutorial, where we have briefly talked about the important problems in the drug discovery and development process, and where AI technologies, especially machine learning technologies, can be helpful, such as a drug molecule property prediction, molecule design, retrosynthesis, drug repurposing with multi-omics data and real-world data, as well as a clinical trial design informed by real-world data, toxicity prediction, and pharmacovigilance. I will, in this part, conclude the entire proposal, entire tutorial with some future directions. So number one is integrative analysis. As we have seen from the contents of this tutorial, so drug development process is really a lengthy process, and which is very, very complex and involves lots of different kinds of data, experimental designs, and machine learning models. This entire pipeline from the initial molecule screening until the final pharmacovigilance, it makes the entire drug development process a holistic pipeline. So we cannot say if we achieve success on a single step or component during this pipeline, then we can get the final success. So it is always important to keep in mind this entire drug development process as a whole. So although in the introduction, we can, in the tutorial, we can see for different components, we pretty much introduce them differently because of the different type of data and different kinds of problems are involved. However, how to effectively and efficiently integrate all the insights obtained by different algorithms and from different data at different steps is something that is really important for the success of drug discovery and development process. And this clearly needs novel methodologies and approaches from machine learning to help with such integration process. Clearly, this is not a simple like data integration or model integration process, but really how can we organically integrate both the data and the model and the insights and make the success rate of the entire process improve. It's an important direction. Number two is model transparency in biomedicine, and especially the drug development and the discovery process. Understanding how the drug works or the hidden mechanism is pretty important, although there are examples like certain drugs that aspirin has been used for a long time without exact knowledge about how, I mean, the mechanism of action, or the underlying mechanism, how it works. But this is based on the pretty broad and wide validation evidence accumulated during the process using aspirin treating patients with different kinds of diseases. So from a computational perspective, it is really important for us to not just tell the domain experts on what we can get, but how we can get that, so that this can improve the trustworthiness of the developed models, and also it can help the domain experts, like the basic scientists or the clinicians, to figure out, I mean, this works or this doesn't work. So what is the reason? And so that they can work together with us, the machine learning scientists, to obtain better models and finally improve the success rate and efficiency of the drug development process. And the third thing is what the so-called model under specification, which is really talking about, I mean, when we really train a really complex model or a big model, we have to like have enough data to make sure, I mean, the model is learned in a sufficient way. But a lot of times, especially in biomedicine, we may not have such large amount of data or samples. This may cause a problem under specification, which states the fact that with the same input and output, there could be multiple routes, which represents multiple different model setups. Those different model setups or parameters can achieve the same output with respect to the same input on your training data, but they can work differently with respect to different testing data. And some of them may cause a pretty bad generalization performance on the testing data because of the distribution drift between the testing data compared to the training data. From this sense, so how can we effectively mitigate the model under specification problem is a promising direction. Like, how can we model the distribution discrepancy between the testing data and the training data, and how can we transfer the knowledge across different data sets, as well as how can we effectively leverage the knowledge from, for example, mechanistic models or from domain experts into the model building process to make it more reliable. And also, how can we reduce the complexity of big models to like build simpler models with fewer parameters. All those are promising directions in the computational drug development process. And the next thing is limited supervision. So we know the drug development process is really a costly process in terms of both time and money. So in order to get supervision for those data, we need domain expert or biological experiments or clinical trials to really give us the ground truth label or supervision information, which clearly cost a lot of time and money. So with the limited supervision information, by combining both the data where has already been annotated with the large amount of unsupervised data is a promising direction to go. For example, how can we leverage like self-supervised learning strategies to come up with better model initializations by pre-training and so on and so forth. So this could also be a very promising direction because we are facing a realistic problem. And the next one is what is so-called the open data science. So because the biomedical data are precious, and a lot of times, especially for clinical data, I mean, the data is typically very sensitive, so a lot of privacy information might be involved. While at the same time, we really want to accumulate or leverage the information from as many data sets as possible to derive collective insights that are more generalizable and trustworthy. So how can we advocate for open data science and data share, such as data sharing, code sharing, and model sharing, is really, really important in biomedicine and especially in the drug development process. So we would also like to call researchers in this field to really, we should join the effort of pushing forward the open data science initiative. And the last one is basically it's kind of like how can we learn in a distributed environment where data are scattered here and there without sacrificing the privacy. So federated learning is really a promising technology that can help us to mitigate the privacy leaking concerns. So it is also very promising technique that can change the game for the computational drug development process. With all that, I would like to thank you everyone for listening to our tutorial. And we have put all the tutorial materials on our tutorial website I put here. And any questions, so feel free to email us. And now we have several minutes for Q&A session.